Our research group is interested in the perioperative inflammation and the relation between inflammatory response to surgery, and mainly the relation between the degree of surgical trauma and the degree of the inflammatory response, and the relation with postoperative complications. In some patients, there's a more pronounced inflammatory reaction after surgery, which is followed by a state of immunosuppression, and this combination is associated with postoperative infections. We know that the type of anesthesia and the administered medications may have an influence on this perioperative inflammatory reaction, and we were interested if there are medications that in a positive sense can influence this reaction, for example by counteracting the postoperative immunosuppression. A brief research in our research group found evidence that sugammadex may have such a potential immunomodulatory effect. And that's why we set up this combined in vitro pilot study and retrospective cohort study. We started with an in vitro experiment, and therefore we obtained peripheral blood mononuclear cells, the so-called PBMCs, from healthy donors. We exposed those cells to various concentrations of sugammadex, rocuronium, or a combination of both. Afterwards, we exposed those cells to a pathogen, and that was in our case the lipopolysaccharide of the E. coli bacteria, and we assessed the ability of the cells to respond to this pathogen. We did this by measuring the cytokine release in response to this pathogen. In our case, we measured the cytokine release of IL-6, IL-1 beta, and TNF. In this in vitro experiment, we found that rocuronium alone had a dose-dependent effect on the cytokine production capacity, and it reduced this cytokine production capacity, and mainly of TNF and IL-6. Sugammadex alone stimulated the cytokine production capacity of TNF, but more importantly, sugammadex after rocuronium seemed to counteract the inhibition caused by rocuronium. As this confirmed our hypothesis of the potential immunomodulatory effect of sugammadex, we continued with the pilot study. In this pilot study, we had 20 patients undergoing total hip arthroplasty under neuroaxial anesthesia, and they were randomized to receive either sugammadex or placebo at the end of surgery. We chose a population of patients receiving neuroaxial anesthesia to exclude the potential confounder effect of other anesthetic agents. To assess the immunomodulatory effect of sugammadex, we obtained blood samples, and we obtained them before surgery, what was the baseline, after administration of placebo or sugammadex, and at postoperative day 1. In those blood samples, we assessed the circulating cytokines and the danger-associated molecular patterns, the so-called DAMs. And in addition, we stimulated those whole blood ex vivo with lipopolysaccharide of the E. coli, and this determine the cytokine production capacity in response to this pathogen. And we measured TNF, IL-6, IL-1 beta, and IL-10. This is a method to assess the immunosuppression, as it is a method to assess what the cells can do in response to a pathogen. In addition, to assess the clinical effects of sugammadex, we used a different cohort because in the pilot study we only had 20 patients, and we already had a retrospective cohort of 1,000 patients of another study, in which we had information on sugammadex use and postoperative complications. So in this cohort, we searched for an association between those two. In our clinical pilot study, we could not extrapolate the findings of the in vitro experiment, so we did not find a difference between the sugammadex and placebo group regarding the circulating cytokines, the concentration of DAMs, or the response to the ex vivo cytokine production capacity. And in addition, we could not find an association between the sugammadex use and the 30 days postoperative complications in the post-hoc analysis of the retrospective cohort. In the clinical use dosing ranges, we could not find a direct immunomodulatory effect of sugammadex. As in the pilot study, we used 8 milligrams per kilogram of sugammadex. That is within the recommended dosing range, but already a bit higher than what is mostly used in clinical practice. But in the in vitro experiment, the concentrations ranged from approximately 2 milligrams per kilogram to a dosing which was way higher than used in the clinical dosage. And the most pronounced effect was mainly seen for the high dosages. And in addition, this illustrates the translational gap between basic research and clinical outcomes. In vitro models, of course, cannot fully capture the complex pharmacodynamics in vivo. But that's why it's interesting that we could combine both methods. So we could combine the in vivo experiment together with the in vitro experiment together with the clinical pilot study. The effect of what we saw of rocuronium in the in vitro experiment does highlight the need for further studies into anesthetic impacts on positive immune responses, because it does show that they do influence this inflammatory response. But yeah, the effect on clinical outcomes is just not known.